Hi, welcome to today's AP Biology lesson. I'm Allison Kate, and before we get going with today's topic, I want to go back, and I promised you the last time we were together that I would give you a correction for some techniques and math errors when I originally presented this chi-square analysis that is used with the mitosis and onion root tips lab during the presentation on Tuesday. So first of all, I presented this slide to you on Wednesday, and this is just the screenshots of the AP Biology lab manual in Lab 7 that shows you the technique of how to calculate the expected data from your observed data. And I originally used a different technique, but I decided I wanted to go back and do it according to the instructions in the lab manual so that you guys can actually go back and have a resource. So what I'd like to do today is apply this to the lab data that I gave you in that original presentation. So you might recall that this was student-generated data, which is observed data for the treatment group lectin. And what you're looking at is the control group in interphase and mitosis, and I have gone back and labeled those cells A and B, and then you're also looking at interphase and mitosis for the lectin-treated group, and I've labeled those cells C and D. And this way you can go back and look at the formulas that are presented in the lab manual. I should also point out that it doesn't affect the end result and the evaluation of the chi-square value. So that said, this is how you calculate your expected results based on the instructions in the lab manual. So the formula, I have TI, which is the treatment group for interphase, and TM, which is the treatment group for mitosis. So for the treatment group in interphase, you're going to take C plus D, and then you're going to multiply that to A plus C. And then you're going to divide that by the total number of cells, which would be A plus D plus A plus B plus C plus D. And then you're going to, for the mitosis treatment group, use cells C plus D and multiply that to B plus D, and again, divided by the total number of cells. So let's just take our data and plug in the numbers. So for the treatment group in interphase, we're going to take 146 and add that to 54, and we're going to multiply that to the sum of 186 plus 146 and divide that by 400. And then for the mitosis treatment, it's 146 plus 54 multiplied by 14 plus 54 over 400, or the total number of cells. And again, we get those numbers, and we end up for the number of cells in the lectin-treated group that are in interphase is 166, and the number of cells that are in the in mitosis in the lectin-treated group is 34. Okay, so now we're going to go ahead and look at the chi-square analysis table. So our observed values in interphase from the lectin-treated group is 146, and for mitosis, it's 54. Our expected cells are what we just calculated. So 166 for interphase and 34 for mitosis. And then we just go ahead and plug those into our chi-square formula, observed minus expected squared over over expected. And the same thing for mitosis, 54 minus 34 squared over 34. If we add the sum of those two results, we're going to get a chi-square value of 11.7. Then we have to recall that we need to compare that to a critical value that we derive from a critical value table in order to evaluate. In other words, to say what that value tells us. So from the critical values table, we're going to be using a p-value of 0.05, which means that we're going to be 95% of our result, and that it is not attributed to chance alone. Our degrees of freedom are the number of categories minus 1. So we have two categories, interphase and mitosis, minus 1. So our degree of freedom is 1. And if we look at our critical value tables, it tells us that our critical value is 3.85. So now we're going to compare our chi-square value, which is greater than our critical value. And that means that we are going to reject the null hypothesis. In other words, our null hypothesis is that there is no difference between the cells in mitosis in any group, right? So there is a difference, so we're going to reject that. And what it actually means for the experiment is that the lectin treatment has more cells in mitosis than we expected. Lectin has an effect on cell division. It increases it. Okay, and that was our alternative hypothesis, which can be confirmed with an accuracy of 95%. So I hope this clears that up. Please feel free to go back. And again, thank you to the students and teachers who pointed this out. All right, so today's lesson is on non-Mendelian genetics. We're going to look at two science practices. We're going to practice with concept explanation and also performing mathematical calculations. And today, they're mostly doing probability calculations and recombinant frequencies to determine map units. So from yesterday, you should be familiar with Mendelian inheritance patterns. You should also be familiar with autosomes versus sex chromosomes. Today, I'm going to use some terms: pleiotropy, epistasis, polygenic inheritance, and multiple alleles, and these all have to do with the non-Mendelian genetic inheritance patterns. By the end of today, you should be able to explain, I just made up a new task verb, you should be able to explain deviations from Mendel's model of the inheritance of traits. So first, we're going to go over our warm-up, which is just a multiple choice question, but it really gives you some practice at thinking about how Mendelian inheritance patterns work. So we have a blue-flowered African violet of unknown ancestry. So we don't know if it is going to be homozygous, dominant, heterozygous, or homozygous recessive. And it self-pollinated and produced 50 seeds. The seeds germinate and grow into flowering plants. Of these plants, 36 produce blue flowers and 14 produce pink flower. What is the best explanation for the pink flowered offspring? So this was your homework from last night. From Ms. Pfizer, which is Mendelian inheritance. And I want you to think about the Mendelian ratios that you learned about yesterday. And then think about that math, because what Mendel was really helpful in doing is providing that mathematical analysis to his observations. So your choices for this are: blue flowers are incompletely dominant to pink flowers, and we're going to talk about incomplete dominance today. Pink flower color is recessive to blue flower color. Pink flower color is the result of somatic mutations in the flower color gene, and a previous generation of blue flowered parent, I should say must have included 50% pink flowers. Okay, so think about what the ratio of 36 to 14 is in Mendelian speak. Okay, so here are the scoring guidelines. The correct answer is, of course, B. Pink flower color is recessive to blue flower color. And sometimes it's a little bit easier to see this if you actually draw out the Punnett square. And so in this case, that blue flower was heterozygous. And so when it combined to produce offspring, it produced 3 to 1, 3 blue to 1 pink. And that's a rough 3 to 1 ratio. Okay. So more practice with this as we go through today. All right, so here's what you need to know. And I know that Ms. Pfizer talked about sex-linked inheritance yesterday, but I want to go over a few other things with regard to sex-linked inheritance patterns, which technically could be placed either in Mendelian or non-Mendelian, but in our course and exam description, they are included in section 5.4, which are non-Mendelian traits. So some traits are determined by genes on sex chromosomes and are known as sex-linked traits. The pattern of inheritance of sex-linked traits can be predicted from data, including pedigrees, which I'm going to share with you in just a moment, which indicate the parent genotype and phenotypes, as well as the offspring's genotypes and phenotypes. So the image that you're looking at on the left is an X chromosome and a Y chromosome. And I really want you to see the difference in size. The X chromosome is much larger than the Y chromosome. And so now if you look at the image on the right, you can see that this is not exhaustive, but there are many, many traits that are carried on the human X chromosome. Compared to the human Y chromosome, there are really only a few traits that are carried on the human Y chromosome. Excuse me. And that is probably the most important one on the Y chromosome is the SRY gene. And that determines during embryonic development, it will determine the male sex characteristics. Excuse me. So most of the sex-linked traits are going to be carried on the X chromosome. And so when we talk about sex-linked traits, we're really talking about X-linked traits. So I want to share with you some ways to visually represent these inheritance patterns. And I'm first going to show you a Punnett square, and then I'm going to show you a pedigree, which are both visual representations of these concepts. So I have 3 different Punnett squares for you. And remember that for an X-linked trait to be expressed, that is for you to actually see the phenotype, a female needs 2 copies, and we call that being homozygous, where a male only needs 1. And we call that being hemizygous, which is kind of a new terminology because really they only have 1 X chromosome. So the gene that's carried on that X chromosome is called hemizygous. And again, you're going to see why X-linked recessive disorders are much more common in males than in females. And that's because males only have that 1 X chromosome. So females, because they have 2 X chromosomes, they can, if they have a dominant allele on the other X chromosome, it will actually not be, the trait will not be expressed in the heterozygous condition. So in Punnett square number 1, we see the female, which has 2 X chromosomes. By the way, in terms of nomenclature, we usually do these crosses with the X chromosomes, and then we show the trait as a superscript up here. So in the first Punnett square, in the first example, we see a female who is not affected. She has 2 X chromosomes with the dominant alleles, whereas the male is affected. He has 1 X chromosome and it has the recessive allele. So that shaded dark shade means they have the condition. Then we can see as the gametes are put across the top and the male gametes and the eggs along the side, and we combine those, we can see that we're going to get all the females, the XX females, are going to be carriers. They're shaded lightly. That means they have 1 dominant allele and 1 recessive allele. And then we have the males, and since they only have 1 X chromosome, they got it from their mom, and they have the normal allele. In the second Punnett square, we see, excuse me again, allergies here in California. We see that the female is a carrier and the male has a normal allele. And in this case, we are going to see that there is a 50-50 chance of the boys getting the X chromosome that has the recessive allele as well as the dominant allele. And then finally, in the third case, if the female is a carrier and the male is afflicted, then we can get, that's the only way we can get a daughter that would have the condition, because she would inherit the X with the recessive allele from her dad, and then the X, she has a 50-50 chance of getting that from her mom. Okay, so those are the 3 different possibilities for what happens if you're going to see an X-linked trait being passed down. There are some human disorders that are X-linked traits. Color blindness, there are some other ways to get color blindness, but it's mostly through this X-linked inheritance pattern. Also a certain type of muscular dystrophy and hemophilia, which is a blood clotting disorder. And I'm going to use that as my pedigree example as as well. So I'm going to do this both ways. I'm going to do it as a Punnett square and a pedigree so you can see the difference. So hemophilia is a sex-linked recessive trait. A female is the carrier for hemophilia and marries a normal male. So we've got our male gametes. He's normal, so he's got the dominant H. Our female is a carrier, so she's got a dominant H and a recessive H. So when we do the cross, we're going to get a normal female, a carrier female, a normal male, and an affected male. And so what's going to happen now is we're going to get some questions, and then I'm going to bring this back to the pedigree. So you might be asked, what fraction of the female children will have hemophilia? And it's really important to read that question carefully because they're asking about the female children, not the male children, not all the children, but the female children. So how much fraction of the female children? None. 0% will actually have the disease. What fraction of the female children will be carrier? You can look, 50% will be carriers and 50 will be normal. What fraction of the male children will have normal blood? 50%. And what fraction will have hemophilia? 50%. Okay, so again, you can do the Punnett square as usual. I always say draw it because it really makes it clear. So now if we were going to look at a pedigree, which is a really common visual representation of these genetic inheritance patterns, we can see, and this is from the royal family, and this is exactly like my Punnett square. Queen Victoria was a carrier, so she's half shaded. By the way, let me do a key first. Males are shown as squares, females are shown as circles, and affected male is shaded, and affected female is shaded. They're not showing you here, a carrier would be half shaded. A line between the male and female represents mating, and then you can see the offspring with the birth order going from left to right. So Queen Victoria was a carrier. And Albert was unaffected. In their F1, their daughter is unaffected, but their son has hemophilia. And then their next born son does not have it. Remember, 50-50 chance. And their daughter is a carrier like their mom. So again, just really wanted to point out the different ways that you can see these types of problems or questions. You could get it as a Punnett square, or a pedigree. All right, so now I want to talk about some other inheritance patterns. We just talked about linked genes. Those were sex-linked genes, but genes can also be linked together on the same chromosome. So genes that are adjacent or close to one another on the same chromosome are linked genes. The probability that genetically linked genes will segregate as a unit can be used to calculate the map distance between them. And I did share this with you when we went over the meiosis lab, but if you didn't get it then, this will be kind of a review. So what we're looking at is a linkage map of some genes in fruit flies, and we're going to do a lot of practice questions with fruit flies, so I thought this would be a good way to start. You can see the wild type phenotypes are on the bottom, and mostly I want to focus on gray body, red eyes, and normal wings, and then along the top, the mutant phenotypes would be black body, cinnabar, and vestigial wings. And you can see that the distances between the genes are what we call map units, and one map unit represents a 1% recombination frequency. So I'm going to show you how we determine that recombination frequency. So the first thing, and this kind of goes back to the meiosis lab, there's evidence that supports the claim that the farther apart two genes are, the higher the probability that a crossover will occur between them and therefore have a higher recombination frequency. So the farther apart those genes are on the chromosome, the larger that number. So this is some data from a test cross in flies, and we can see that we have 965 wild type, 944 black vestigial. So we cross the two homozygous recessives. But then we see some new phenotypes. We get a gray vestigial, 206, and 185 black normal. Now, the first two we call parentals because they look like the parents in the cross. The second two we call the recombinants because they do not look like the the parents that were used in the cross. So to determine the recombination frequency, we take the total number of recombinants, which is 206 plus 185, or 391 recombinants, and we divide that by the total number of offspring, which in this case is 2,300. We multiply it by 100 to get a percent, and you can see that the recombination frequency is going to be 17%. That means that the genes for body color and wing length are 17 map units apart. Okay, so let's take a look at some other ways that traits are inherited that are not typical Mendelian inheritance patterns. And I want to talk about a few of them. So this is the overview, and I will end with a review slide. So we're going to talk about the levels of dominance, incomplete dominance, and my examples are going to be in roan cattle and flower color, and I'm showing it another way for nomenclature purposes. And then we've got pleiotropy, where one gene has multiple effects on the phenotype. Sex-linked inheritance, which we just discussed. Non-nuclear inheritance, which means that there are some organelles that have their own DNA, and they can be inherited separately from genomic DNA in the nucleus. Polygenic inheritance means that there are many genes that influence the phenotype. And epistasis, which is when one gene will have an effect on the expression of another gene. Okay, so let's talk about the degrees of dominance that we're going to see in this example. So first of all, complete dominance is the Mendelian inheritance pattern, and it occurs when the phenotypes of the heterozygote and dominant homozygote are identical. So if this was a cross between a red and a white flower, and we would see a heterozygous genotype, it would still be a red phenotype. In incomplete dominance, the phenotype of the F1 hybrids are somewhere in between the two phenotypes of the parental varietals. So in my example, I'm crossing a red snapdragon with a white snapdragon, and in the F1, we get a pink flower. And this is not blending. And the reason that it is not blending is that if you then cross those two F1 pink flowers, we can see that the original phenotypes do appear. So we will get a 1 red to 2 pink to 1 white. And this is going to be different than the 3-to-1 Mendelian pattern. Some examples of this, of course, flower color, roan cattle, and blue chicken. So if you see a question about any of those kinds of examples, I want you to think incomplete dominance. Okay, remember that the ratio is 1 to 2 to 1 in the F2, not the 3 to 1 that you learned in the Mendelian ratios. And then the last degree of dominance is codominance, where two dominant alleles affect the phenotype in separate and distinguishable ways. And I'm going to give you an example in blood type in just a minute, but just to tell you the difference in flowers, right? If you did snapdragon flowers, you would see equal numbers of red and white flowers. They would be expressed the same on the same plant rather than seeing that pink flower. So I'm going to use blood groups for 2 examples. I'm going to use it for multiple alleles and codominance. And you may or may not know this, that in human blood type, there are actually 3 alleles that code for the different phenotypes that we see in blood typing. So the alleles are IA, IB, and then lowercase I, which will represent the recessive. The IA and IB are co-dominant, which means they will be expressed equally and create a new phenotype. So what's really going on when we talk about these blood types is that on the surface of your red blood cell, you have a gene that will code for a carbohydrate marker. And if you have IA, you are going to have the carbohydrate A on the surface of your red blood cell. If you have the gene IB, you're going to have the carbohydrate B on the surface of your blood cell. If you have both of them, IA and IB, then you'll have a new phenotype, AB, where both of those carbohydrates are expressed equally. And if you're recessive, you don't have either of those carbohydrates. So you have it's a lack of carbohydrates. I should also go back and tell you that to get blood type A, you can either be homozygous for A or you can be heterozygous AI. Same is true with B. You can be homozygous for B or you can be heterozygous BI. Now, I'm not going to go into detail right now about blood compatibility. That's kind of a different lesson, but you can be thinking about how that might work. So just to review, most genes exist in populations in more than 2 allelic forms. In this case, we have 3 alleles, IA, IB, and lowercase I. And again, the I gene is coding for an enzyme that adds a specific carbohydrate to the surface of the blood cells. And so you're either going to get A, B, both A, B, and both A and B because it's codominant, or none, which means you have blood type O. The next non-Mendelian inheritance pattern I'd like to talk about is called epistasis. That word literally means to stand upon. So in this case, epistasis is going to have 2 different alleles that are going to, one set of alleles is actually going to change the way the phenotype is expressed. The common example that's used is in labs. And if you have a lab, you know that they come in different varieties. We have black labs, chocolate labs, which are brown labs, and then we have yellow labs. And yellow labs can actually be 2 varieties. Their fur color is yellow, but then the other areas that are pigmented, like their nose and their eyes and their paws, may vary in the amount of pigment that they get. So in epistasis, a gene at one locus will alter the phenotypic expression of a gene at a second locus. And we say that one of the genes is epistatic to the other one. And so the 2 genes that we're talking about in labs is that there's a gene that determines the pigment color, which is going to be dominant for black, recessive for brown, and then another gene that we're calling the C gene, which is for color or no color, and that determines how much pigment will actually be deposited in their fur or hair. And so you can see we've got 2 this is a dihybrid cross, and one of the ways that you can tell epistasis is that it is not a 9:3 to 3:1 ratio, which is what we would expect if we were doing a Mendelian dihybrid cross, right? So in this case, it's modified to a 9:7 or a 9:3:4 ratio. So if you see that on an exam question, you'll immediately want to think epistasis. And so you can see that there are going to be, in this dihybrid cross, 9 dogs that are going to have black fur, and it's either because they're dominant for the black allele, right, like here and here, or they're homozygous they're heterozygous for the black allele. In order to be a chocolate lab, you have to be homozygous recessive for the color allele, for the pigment color allele, not the amount of color. And then in these 3 cases, what is happening, actually, let me start here. So this is just our recessive, our homozygous recessive, and that's the one that we would expect to be light fur, right? So that would be our 9:3 to 3:1. Now these 3, what is happening is the amount of pigment, the amount of color is the homozygous recessive. And even though we would expect it to be brown because we've got the homozygous dominant and heterozygous genes for color, because we get this homozygous recessive in the amount of color, we are going to say that it is epistatic to that brown pigment, and so we don't see that being expressed. So it actually stands upon the color, and instead of getting a chocolate lab, we are going to get a yellow lab. I hope that made sense. Okay, so pleiotropy. Pleiotropy is a funny word. Basically, it means that one gene has many different phenotypic effects, and there are many hereditary diseases in humans, such as cystic fibrosis or sickle cell disease, that have this. There's also a weird trait in chickens called the Frizzle feather trait, I think, and it affects the defect in the gene affects the cartilage. So anywhere that the cartilage is in the chicken, such as in their feathers or in where their bones connect, the ends of bones, the cartilage is affected. So they can't fly, the feathers don't provide any thermoregulation, and they're deformed. And so, you know, that's important in the pecking order of chickens. So having this one gene defect affects a lot of the phenotypes for that chicken. So again, pleiotropy is one gene, has many phenotypic effects. And then I think this is my last non-Mendelian inheritance pattern. It is polygenic inheritance. Poly means many, genic is gene. So it is an example of the additive effect of two or more genes on a single phenotype. And there's lots of examples of this. It's anything that is along a trait that's along a continuum, like height or skin color. And in other organisms, it might be things like the seed, number of seeds or seed weight, that kind of stuff. So in this case, many genes, there are 6 alleles that are going to actually be responsible for skin color in humans. And the easiest way for me to explain it is if you were to receive all 6 recessive alleles, like in this first square, you would have the lightest possible skin color. If you were to receive all 6 of those dominant alleles, then you would have the darkest possible skin color. And then to get this range that we see in human skin color, it could be any of these different possible 64 possible combinations that would give you a different phenotype. And this is just the genetic side. And just to throw in the effect of environment, so let's say that you're here and you have all of 6 recessive alleles and you have very light skin color. If you are out in the sun, let's say you take a summer job where you're out, you know, a lifeguard, or you're working on trail restoration, you might be out in the sun and exposed to that ultraviolet radiation, which is going to stimulate your melanin production. And even though genetically you have light skin, the environment can change that color. So genetics is complicated, and it also has an interplay with the environment. So just a quick review of all of the non-Mendelian inheritance patterns that we just talked about. So just to get the range of dominance, complete dominance of one allele over another is the Mendelian inheritance pattern. You have incomplete dominance, which the classic example is seen in flower color. Don't forget your roan cattle or your blue chickens, which are actually gray because the two phenotypes are black and white. They just look blue. Codominance, where you see both phenotypes expressed equally. Blood type AB is our example. Multiple alleles, where we also use blood types as our example, and those alleles are A, B, and I, lowercase I, the 2 dominants and the recessive. And then pleiotropy, where one gene is able to affect many phenotypic characteristics. You can think of sickle cell or that Frizzle trait in chickens. Epistasis is where the phenotypic expression of one gene affects the other gene. And polygenic inheritance, where a single phenotypic character is affected by 2 or more genes. All right, let's get some information about the last type of non-Mendelian, and this is traits that result from non-nuclear inheritance. And you might recall from previous lessons that both chloroplasts and mitochondria have their own DNA. You might recall that it is very similar to prokaryotic DNA in that it's circular, like the plasmids that are seen in bacteria. And you might want to think about the evolutionary reasoning behind this and the endosymbiotic theory. But basically what happens in the way that mitochondrial DNA in animals and chloroplasts and mitochondria DNA in plants, the way that it is assorted to gametes and daughter cells is through the maternal lineage. And I'm going to give you some examples. But if you look at this picture, you can see that the nuclear, there are 2 copies, and you inherit it from each parent. So the red X maybe came from dad, and the black X came from mom. The mitochondrial DNA, which is cytoplasmic, only comes from the mother. And the reason for this is that when a sperm cell fertilizes an egg, really all it's providing is its nucleus. It doesn't provide any of the cytoplasm. All of the cytoplasm comes from the maternal, the ovum. Okay, so In animals, mitochondria are transmitted by the egg and not the sperm. And so what you see is over here in my pedigree, it's a different way to show a pedigree. If the mom has some type of a trait that is passed down through mitochondrial DNA and she has that trait, all of her children will get it, because whether you're a girl or a boy, you're going to get an X chromosome from mom, right? Dad, doesn't matter because there is no DNA coming from the dad's nucleus. On the other hand, if the dad is not affected and the mom is unaffected, wait, did I say that wrong? The dad is affected and the mom is unaffected, all of her children will also be unaffected. And again, this image shows you a circular piece of DNA from the mitochondria. And most of those genes really have to do with providing proteins that are used in cellular respiration. Same thing is true in chloroplasts, but this is in plants only, right? So they get both mitochondria and chloroplast DNA, which is transmitted in the ovule, which is the female gamete, and not in the pollen, which is the male gamete. So again, this is a nice illustration. The male, the pollen, does not have the chloroplast DNA or the mitochondrial DNA. So when it fertilizes, it only brings the genes that it has in its nucleus, whereas the ovule has both chloroplast mitochondrial DNA in addition to the genes that are in its nucleus. Okay, so let's get some practice with these concepts. The first one, and these I know take a lot of reading. I'm going to go through this and then I'm going to give you the answer in words and then I'm going to show you in some diagrams as well. So what you're looking at is a data table of the phenotype of some F2 flies. So let's look at who was crossed. Each student in the class performed a parental cross between a fly that is true breeding for ebony wings and vestigial wings, and a fly that is true breeding for gray and long wings. And I hope you remember that when I showed you those traits, both gray and long are dominant, and ebony and vestigial are recessive. So we've got parentals that are homozygous dominant and homozygous recessive. And that means that our F1 flies are going to be heterozygous for both of those genes. Then the student crossed several pairs of F1 flies and determined the phenotypes of 500 of the resulting F2 flies with respect to body color and wing shape. The students in the class averaged their data for the frequencies of the 4 possible genotypes. And when I'm going through this, I want you to be thinking about why it's important to have those numbers, and think about Mendel and what his great contribution to the study of genetics was. It was being able to apply mathematics and statistics to the observations that he was seeing in his crosses with pea plants. So that number 500, those numbers that you see in the data table, ebony body and long wings is 98, ebony body and vestigial wings is 28, gray body and long wings is 293, and gray bodies and vestigial wings is 81. So based on this data, describe why the dominant alleles for body color and wing shape are the alleles that produce gray body and long wings. Okay, and this is 3 parts. And then based on that data, describe why the 2 genes are most likely on different chromosomes or why they are on the same chromosome. So in other words, are they linked or not linked? And then you're asked to calculate the probability of producing flies that have gray bodies and vestigial wings if a cross is performed between one of the F1 flies, okay, so the hybrid, the dihybrid, and the first analysis, and then a fly that is homozygous, which is homozygous dominant for gray body and gray body and vestigial wings is the recessive. So that would be homozygous for the dominant trait and then homozygous for the recessive trait. Okay, so you're going to have to work that out, but I'm going to show you. All right, and then of course we've got our plus and minus 2 standard errors at the means. Okay, so first of all, if we were to do this, we know that if it was a Mendelian inheritance pattern of chromosomes that, or of genes that are on separate chromosomes, we should see a 9 to 3 to 3 to 1 ratio. And hopefully you looked at those numbers. We have 500, which is a lot, right? And you can kind of see the 9 to 3 to 3 to 1 ratios. Okay, and then probability, the probability is going to be 0.5. And the reason for that is the probability of having gray bodies with the parentals in that cross is 1, and the probability of having vestigial wings is 0.5. So if we multiply 1 by 0.5, we're going to get 0.5. Let me show you that in pictures. So here's the cross, right? So we started off with parents that were a homozygous recessive for both traits crossed with a homozygous dominant for both traits. So our F1 is a dihybrid. If we go ahead and do that Punnett square and we work out, you can see that the boxed genotypes represent the 293, and that's our 9. The circled genotypes represent our, let's see, big E, little v, 81, which is our 3. The triangles represent our little e big V, which is our 98. And then finally, our little e little v, which was 28, is 1. So the 9:3 to 3:1 ratio of the F2 phenotypes supports the claim that the gene for body color and wing length are on separate chromosomes. To do the probability calculation, the cross was our F1, right, which was the dihybrid, big E, big V, little v, and then the dominant for the gray body type, big E, and the vestigial for the wing, little v. So if you do them separately, you could obviously do a shortcut, but if you do them separately, I just wanted to show you how it came out. For the body color, they're all gray, so that probability is 1. For the vestigial, you get half long, half vestigial, so that probability is 0.5, and then the overall probability is 1 times 0.5. Oops, that should be 0.5 right here, sorry, but the correct answer is 0.5, okay? All right, let's get some practice in plants because plants are important as well. So in plants, genes involved in chlorophyll synthesis are located in both the nucleus and the chloroplast. Some variegated plants with both green and white leaves and shoots have mutations that prevent them from synthesizing chlorophyll throughout their leaves. In an effort to understand the genetics underlying chlorophyll synthesis, scientists performed the crosses shown in Figure 1. So what I want you to see, this is the outcome of the leaf, right? So you can have green, you can have white, and you can have a combination of green and white, which is called variegated. So in this case, when the ovule is green, the progeny is green. It doesn't matter what the pollen that fertilized that ovule is. It could be green, white, or variegated. The progeny is green. Ovule is white, same deal. We get the progeny is white regardless of the pollen. In this case, when the ovule is variegated, again, it doesn't matter. We're going to get variegated. Okay. So, 4-part question. Describe how genetic information is carried in chloroplasts and mitochondria. Okay, got that? We know that chloroplasts and mitochondria contain their own genetic material. B, explain how the inheritance pattern that's shown in Figure 1 supports the role for chloroplasts supports a role for chloroplast genes in the production of chlorophyll in the variegated plants, right? So we know that the chloroplasts are only transmitted to the offspring, the progeny, from the female parent, which is the ovule, and that the progeny produced always matched or can be produced from the ovule used in the cross. So you just need to, I don't know if I can go back, whoops. You can just see that it doesn't matter what the pollen is, whatever is in the ovule, the maternal parent, that's what the offspring are going to look like. Okay, so now ovules produced by an entirely green plant contain a rare mutation, keyword here, in a nuclear gene that is involved in chlorophyll synthesis, the gene that encodes a non-functional protein. Predict the most likely color of leaves on plants that are produced when these ovules are fertilized by pollen from a wild-type green plant. Okay, so now you're asked to shift gears and think about a mutation in a nuclear gene. What color are the offspring going to be if the pollen was from a green plant? Okay, and the leaves are going to be green. This is a nuclear gene that has a mutation, not a chloroplast DNA gene. And the reason for that is we need to justify is that the plants will inherit the functional gene from their dad, from the pollen, in the wild type green plant. So depending on the amount of the genes encoded protein that's needed for chlorophyll production, the leaves might be paler green, but they will all be green because they got that gene from the pollen. All right, quick check for understanding. I'm going to try and do this quickly, and then you can practice with this later on. Okay, so again, these take a long time, and they really require that you do some careful reading. Fruit flies in Drosophila melanogaster with a wild-type phenotype have gray bodies and red eyes. Certain mutations can cause changes to these traits. Mutant flies have a black body and are cinnabar eyes. To study the genetics of these traits, a researcher crossed a true breeding wild-type male, which would be gray body and red eyes, with a true breeding female fly with black body and cinnabar eyes. So again, we've got a homozygous dominant with the homozygous recessive. We get our F1, which are all heterozygous for each trait, so they're dihybrid. Female flies from the F1 generation were crossed with true breeding male flies with black bodies and cinnabar eyes. So now we're crossing the F1 hybrids with a homozygous recessive male for both traits. The data on the next page, it actually is not on the next page, it's right there in front of you, represents the predicted outcomes and the data obtained from the cross. You are asked to explain the difference between the expected data and the actual numbers observed. So think Mendel, if these genes were on the different chromosomes, right, we would expect 1 to 1 to 1 to 1. So we get a same number, 244 for each of the F2 phenotypes, but that's not what happened. We got an observed number of the parents. So we have a gray and red, right? So that was one of the parentals. We had a black and cinnabar, which was the other parent in that first cross. And then we have two new phenotypes, gray and cinnabar and black and red. So we call those the recombinants. So now you need to explain that difference. Okay, so your explanation needs to include the idea that we would predict a 1-to-1 ratio if it was Mendelian inheritance pattern. You could do a Punnett square that shows that cross, the dihybrid parent with the homozygous recessive parent. A correct application of the chi-square analysis to show that the observed results do not conform to the expected frequencies. In other words, these are not 9 to 3 to 3 to 1. You could say that because of those things, the body color and eye color are linked genes. You could show ratios to show linkage and independent assortment of wing type versus linked traits. You could identify the bottom two phenotypes as the products of crossing over, which are those recombinant phenotypes. And then you can also do the crossover rate. You could do the recombination frequency, which is between 9 and 10%. So again, let me show you working it out. You can see the F1 dihybrid crossed with the homozygous recessive. We should get a 1-to-1 to 1, but we did not. So why? The why is that the eye color and body color genes must be linked. And you can actually show that with math by calculating the recombination frequency by adding 42 and 47 to get the 89 recombinants out of the total number of offspring, and that's about a 9% recombination frequency. All right, so to debrief, you should now be able to explain deviations from Mendel's model of the inheritance of traits. You should be able to explain biological concepts and perform some types of mathematical calculations. Your key takeaway, as it's been all week, is that heritable information provides for the continuity of life. I want to leave you with some common errors and misunderstandings. This question was given, might have even been on 2018 AP Biology exam. And this also shows the effect of the environment on the expression of the genotype as phenotype. So this is kind of a funny one. In the tongue soulfish, its name is cynoglossus. Glossus means tongue. Sex is determined by the combination of genetics and environmental temperature. Genetically male fish have 2 Z chromosomes, ZZ, and genetically female fish have 1 Z and 1 W, they're ZW. When fish are raised at 22 degrees C, ZZ fish develop into phenotypic males and ZW fish develop into phenotypic females. However, if these fish are raised at a higher temperature, 28 degrees C, the Z chromosome is modified. We denote it as Z star. Z star W individuals develop as phenotypic males that are fertile and can pass on that Z star chromosome to their offspring, even when the offspring are raised at 22 degrees. They're showing a cross between a ZW female and a Z star Z male in that Punnett square. You are asked to predict the percent of phenotypic males among the F1 offspring of this cross if the offspring are raised at 22 degrees C. Then you are given another task, which is that if at least one Z or Z star chromosome is necessary for the survival of the fish, a researcher crossed two fish and observed a 2-to-1 ratio of males to females among the offspring. Based on the information, identify the genotype of the male parent in the cross, and then finally describe one fitness cost to the female of mating with this particular male. Okay, so here's the scoring guidelines. The prediction is that there would be 75% males, and that's part A. And then in part B, the 2-to-1 ratio of males to females means that you would have to have the male parent being Z star W. And the reason that there's a fitness cost, as you'll see when I show you the Punnett square, fewer offspring will survive. The individuals that get the WW genotype will die. Okay. And another thing is that all of those male offspring will have the Z star chromosome, and that doesn't bode well for the next generation. So if you look at this cross, in the first part for your prediction, you've got your ZW female and your Z star Z male. 1, 2, 3 males, or 3 of those offspring are male, so 3 out of 4 is 75%. The only way to get a female is ZW. To make that prediction, here's the Punnett square. Remember, we get a 2-to-1 ratio, so one of the offspring actually died, and that is that WW genotype. So the parent is Z star W. So I'm just going to look at these results. So the good news is that most students were able to use the Punnett square to correctly predict that 75% of the offspring would be phenotypically male. In part B, there was a drop-off. Most students could identify the likely genotype of the male parent as Z star W. However, only some students were able to discuss the fitness cost to the female, in that she would have fewer offspring that survive, or that some of her offspring would have that Z star chromosome. Okay, so here's your homework. We've seen this one before, but it's a different part of this question. You have a cell, and there are 2 chromosomes. Chromosome 1 is for the color of the flower, either green dominant or purple recessive, and chromosome 2 is for plant height, either dwarf, which is dominant, or tall, which is recessive. So the individuals of the parental generation with the genotypes of big G, big G, big D, big D, and little g, little g, little d, little g were crossed to produce F1 progeny. Now here's your new question. If the 2 genes were genetically linked, describe how the proportions of the phenotypes of the resulting offspring would most likely differ from those of the test cross between an F1 individual and a GG/DD individual, homozygous recessive. Okay, so work that out. Basically, you're wanting to answer, how can you tell the difference between if genes are linked and not linked? All right, that is due on Monday because it's the weekend, and Ms. Pfizer will be with you on Monday to go over the answer to that. Just want to remind you of your exam checklist. Today is the 24th of April. It's the last day to reach out to College Board. If you need a device or internet connectivity, please reach out to them at cb.org/tech. Also, check to make sure that the College Board has your correct email address. They will be sending you your exam ticket via email, although you will be able to access it through AP Central if you do not receive the email. And then finally, let your AP Bio teacher in on your plan to test or not to test. Okay, so thanks for watching. We hope that you'll join us again. Make sure you complete that practice question for Monday. And Monday's lesson will review chromosomal inheritance and the environmental effects on phenotype. Thanks for watching. Have a great weekend. See you guys next week.